but specifically the circular DNA, which is how AAV is associated with long-term expression. We learned in the early days that we discovered like AAV8, AAV9, whatever, that was much better than AAV2, that we found a peak of expression in week 2, but then the expression goes down a little bit and reach the plateau. The idea behind that was that initially, when you inject the vector, it's not self-complementary, it's just single DNA. All the single strand together with the double strand express the transgene. But after 2 weeks, the single-stranded is not very stable. They tend to be transient. So therefore, we thought that the upper regulation of expression 2 weeks is a combination of the circular plus the single-stranded. But over time, the single-stranded is not very stable, and the circular takes place. However, this takes like 1 or 2 weeks after the vector injection. And some animal models reproduce that to a certain extent. However, we have not seen this take a wait for 2 years to have this change in the AAV molecular mechanism. And I think it will be very hard to prove unless they do like a very comprehensive non-human primate study or dog study. But from dogs, I can tell what we inject, even not very good vectors that we have the first generation. After 10 years, they're pretty much stable. So it's something unique. And what is the main difference? It's not AAV, it's not serotype, but how you make your vector. And this probably will be something different because all of our study and most of the published study is AAV that was produced in mammalian cell line. Whether this production in the insect cell line use the baculovirus system is different, that I don't know. And unfortunately, I'm very embarrassed to say the field doesn't have one side-by-side comparison. If you take the same serotype, the same transgene, whatever the transgene is, and if you make this in the mammalian system, or if you make this in insect cell line with baculo, are they comparable? 2 years ago at the ASGCT, there was a poster showing that post-translation modification, if you make in a mammalian cell or insect cell line, they are different. Therefore, it is not the same product. So therefore, this data could be related to manufacturing.
So, Dr. Arruda, when you say, the poster, can you elaborate a little bit, how they different in terms of product?
Well, you can go from a long way, like sugar. You can go to, any other activity, if you will. I don't have the poster in front of me to remember this, but you can look at, and they're not the same vector. They are, and it makes sense to me, especially if you think glycosylation, for instance. It depends on the cell line you use. So every glycosylation will follow that specific cell line, and not a general. And it doesn't mean that they're different. I'm not saying that they're bad. I say they're different, but again, it's not the same product. Therefore,
so when you say different,
the best way to address this is because BioMarin has AAV5 in insect cell line. If you look at uniQure early data for hemophilia B, they have also AAV5 and insect cell line. And now we have the Sangamo that has a hemophilia A program, which they have AAV6 insect cell line. It's the first time that we have a different serotype, and look like it doesn't matter the serotype. Look like they need more vector if you use the insect cell line using Bacillus system. Because initially I thought this is a combination probably of AAV5 plus how you make the vector. But when the AAV6 came from Sangamo, they still need a lot more vector than if you compare with, we talk about E13, like 3 to 6 E13 from BioMarin or Sangamo for factor 8, in contrast with the mammalian system that you can go to a log scale lower. So these are different products.
Mm-hmm. Okay. So, I mean, one way is the different, dose, that could that be due to, empty versus full capsid? Other than that, what other differences you see is that, and do you see, I think the main question for BioMarin is like a wide decline, and now, basically stabilize, and then should we expect the stabilizing or like a slower decline maintained going forward? I think that's maybe the main question for us, for the durability question. Yeah. And then, BioMarin certainly did some BioMarin, I think in one of their, quite a few slides, they did a simulation, based on the decline kinetics, and they expected, the 8-year durability above 5% factor A level. And do you agree with that?
Well, I don't. I think because they have such a unique system that only time can tell us. I don't think anybody can guess where this will, reach a plateau. Because, as I said, in the field of gene therapy, we never have this thing that after 2 years the expression goes down. I don't remember any case related to that for a replacement therapy, but it's something unique, and only time is going to let us decide that. And I don't think that their argument is based on solid evidence. It's just their argument. And I think is something that they need to prove. I don't think they have anything published that I can comment about that say, well, this is what it is or predictable. But and I think as a company, they try to come up with explanations and things like this. But no, I'm not comfortable, because the problem, as I said before, we are talking a disease that the life expectancy is 6 decades or longer. Therefore, we have to be pretty sure that this is stable. And right now, I don't think one can tell that, okay, they drop it, and after year 3, the plateau levels will remain the same. This is just a guess. I don't think anybody knows the answer for that, to be honest.
Mm-hmm. So why do you? Yeah, I think that's helpful. But then question is another more scientific question. Why year 3? Now we do see a slower decline. Like from year 1 to year 2, basically drop like over 50%, right? And then year 3, you basically drop 24% if we're using chromogenic assay and looking at the median factor A level. So they drop at 24%. So what could be the reason causing that slow dropping? From like scientific or mechanistic point of view, what could make that happen? And then would that maintain, or would that? So like if you can share with us, your thoughts on your maybe hypothesis or speculation that what could happen.
Yeah. I will go for the old concept that we have is like a dogma in the field, is like cells don't like to make factor VIII for some reason. And this has nothing to do with the size, because they can make factor V in the same way, and factor V and factor VIII, they're pretty much came from the same ancestor gene. But there's always this concept that probably cells will go to ER stress, and therefore this will compromise the folding of the protein and how the protein is secreted. Could be one hypothetical hypothesis, like the cells make, because when you inject AAV in the liver, it doesn't mean that it goes through all the liver. Usually they go to a small part of the liver. I would say guess 20 to 40%. But if you think about that, when you are expressing normal levels, like 100% or even more, it means that few cells are actually overloaded with the vector. They're not like the one gene copy number. They express a lot, and could be that over time some of the cells will start to have problems in secreting factor VIII properly. But again, in my impression is always that this should be earlier, not that late, but remains to be determined. I don't think nobody knows the answer for that. We need to come up with the preclinical models that help us. Mouse